Dear guests, welcome to this academic ceremony on the occasion of David Barnett's PhD defense. On behalf of Maastricht University, I would like to thank you for showing up on this dark and rainy day. But inside we have the light of science and the light of knowledge that will hopefully illuminate us. This is a formal academic proceeding, but at the same time it is also a celebration of all the work that has been done to produce this little booklet, which seems so simple and so manageable, but no doubt is the result of many years of hard work. So while we will ask serious questions and observe a number of ancient rituals and customs, I always like to point out that this is a festive occasion and we should enjoy it as such. And with that, I hereby open this academic ceremony in which David Barnett will defend the academic thesis entitled Perturbing and Protecting the Infant Gut, Exploring the Impacts of Antibiotics and Oligosaccharides on Gut Microbiota and Child Health. May I invite you to present a summary of your study and the conclusions of your thesis.
Thank you, Pro-Rector, and good afternoon, everyone. Dear colleagues, family and friends, dear esteemed members of the Assessment Committee, thank you all very much for taking the time to be here today. I will indeed begin with a short presentation about my PhD research, and I will start that with a short story to introduce the topic. So, before birth, you are isolated from the rest of the world, protected inside your mother's womb. But when you are born, you enter a world shared with other organisms. Some of these organisms will be other humans, like your mother and your father. But most of the organisms that you will meet are microorganisms. And that's because if you could look with a microscope at almost any surface around you, you will find microbes living there in many different varieties. But one place that microbes particularly like to live is in your intestines. And so right now in your large intestines, live many millions of different microbes, cheerfully feeding on whatever you ate for lunch earlier. But this complex ecosystem of microbes took time to develop, and this process starts in infancy. So when you were a baby, growing, developing, changing over time, so too was the ecosystem of microbes living in your intestines, your gut microbiota. This might sound a little concerning at first, but actually many of these gut microbes perform useful functions. Some can help us extract additional nutrition from our food, and some even produce substances that we suspect play important roles in promoting healthy infant development. But the ecosystem in your intestines is different from the ecosystem inside the intestines of the person sitting next to you. And these inter-individual differences in gut microbiota composition already start appearing when we are babies. And this story leads us to two broad research questions. Firstly, what explains these gut microbiota differences that appear during infancy? And secondly, how might these differences be relevant to child health? But those are very broad research questions, and the more specific focus of my thesis research is described in its subtitle, Exploring the Impacts of Antibiotics and Oligosaccharides on the Gut Microbiota and Child Health. Antibiotics are like a double-edged sword. They are useful for treating infections, but they don't just kill bad bacteria. They can also cause harmful perturbations across the whole gut ecosystem, including the sorts of bacteria we think are beneficial to a child's health. Oligosaccharides, on the other hand, may have an important role in protecting healthy infant gut microbiota development. But what are these oligosaccharides? They are special types of carbohydrates or sugars, and human breast milk is particularly full of these special sugars. We call these ones human milk oligosaccharides, or HMOs for short. But HMOs are not food for the baby. They can only be digested by some of the baby's gut bacteria. And we suspect that these HMOs convey some of the health benefits of breastfeeding by promoting good bacteria and suppressing bad bacteria. But there are many different types of HMOs present in breast milk, and we're not sure of all of their different functions. So breast milk always contains some oligosaccharides, but what about formula milk? Are there any oligosaccharides there? Sometimes. Some formula milk products have oligosaccharides added to them, but not all. And one type of oligosaccharides, now added to many modern formula milks, are the galacto-oligosaccharides, or GOS for short. Okay, so now you know a little bit about what oligosaccharides are. I can continue to give some of the highlights of our research. How did we investigate the effects of oligosaccharides and antibiotics on the gut microbiota and child health? And what did we find? So starting with chapter 3, here we looked at over 1,000 babies participating in the koala birth cohort. The parents of these babies had collected baby poop samples at 1 month after birth and completed many questionnaires. And these questionnaires asked about things like how the baby was born, whether they received antibiotics, how the baby was fed, as well as many other questions. But we found that these 3 things in particular could each explain a big part of the differences we saw in the baby's gut microbiomes. Several further factors could also already influence the 1-month-old microbiome, but to a somewhat lesser extent. However, despite all of these influences, there was still plenty of gut microbiome differences that we could not explain. And so next, we looked in more detail into differences in dietary oligosaccharides within 2 different subgroups of koala infants. We looked in data from the 111 exclusively formula-fed babies and saw evidence that those babies who received galacto-oligosaccharide supplemented formula had a somewhat more favorable gut microbiota composition relative to infants fed other types of formula milk without those oligosaccharides. For 220 of the breastfed babies, we also had additional measurements about what types of HMOs were present in their mother's breast milk. And in this group, we found large variation in exactly which types of HMOs were present in each mother's milk. But we found that all of these variations seemed to be similarly beneficial for a breastfed infant, with this breast milk HMO variation not causing any obvious variation in the microbiomes within this group of breastfed infants. So, these HMO things seem to be great, but breast milk contains more things than just HMOs. In another study reported in Chapter 4, we recruited 23 mother and infant pairs who provided breast milk and fecal samples at 1 month, 3 months, and 9 months of age. With this, we investigated, amongst other things, whether microbes present in breast milk might be an important source of microbes for colonizing the infant gut. And we found some evidence to suggest that breast milk microbes might be more important for this at 1 month than at later time points. For our research purposes, though, the baby carb study actually served two purposes. Firstly, as this small cohort in which we could follow the infants and their microbes over the first year of life, but also it functioned as a convenient way to collect baby poop for use in laboratory experiments. So in the experiments described in Chapter 7, my colleague Martha Endicott took some of the samples full of gut microbes from the babies at either 1 month or 3 months of age and put them into a computer-controlled model of the large intestine, the TIM2 model. And in this experimental system, we added not just the gut microbiota from the poop samples, but we could also add antibiotics to purposefully disturb the infant microbiome and see how they responded, something you obviously couldn't do with the microbes still inside the actual babies. But we did not just aim to disturb the microbiome, we also tried to protect the microbiome by adding one of two different types of oligosaccharides to the experiments. Either galacto-oligosaccharides, the one often added to formula milk, or 2-fucosyl lactose, which is a type of human milk oligosaccharide. We found that both of these were able, at least partially, to protect some of the important activities of the microbiota, namely the production of some substances called short-chain fatty acids that are beneficial for gut health. But we also noticed that the response of the microbiota was different, firstly depending on which oligosaccharide we added, but also which age of infant microbiota we tested. Whether it was 1 month old or 3 months old, that also mattered. But okay, now zooming back out from the details to the bigger picture, so far we've looked at the question of what explains infant gut microbiota differences. And next, I'll briefly outline some of our work investigating the relevance of the gut microbiota to child health. In Chapter 5, we investigated infant gastrointestinal symptoms that many parents report seeing in their child. One common symptom was colicky crying, defined here as repeatedly crying for over 3 hours per day for no obvious reason. Parents also often reported that their baby showed signs of either constipation or of stomach cramps. And so, in Chapter 5, we set out to investigate whether gut microbes might be involved in some of these common gut symptoms. To answer this question, we returned to the 1,000 infants from the koala study with their poop samples at 1 month of age. In this cohort, we found that the infant gut microbiota was associated with both the colicky crying and constipation symptoms. And our statistical analyses identified different associations with these different symptoms, specifically with either higher or lower levels of different types of gut bacteria. And one association that I found particularly interesting was the association we identified between colicky crying and a microbe called Ruminococcus gnavis. So this microbe has been associated also with gastrointestinal disease in adults, and it's known to be able to make a type of carbohydrate that could cause harmful inflammation. However, Ruminococcus gnavis is also found in the guts of many healthy individuals without gastrointestinal problems. So, as is often the case in science, the story cannot be quite so simple. So, in the last few minutes, I've been telling a simplified overview of the story of my PhD research, intentionally leaving out some of the more complicated details. And one complicated part of microbiome research is making sense of the data that we obtain from the complex microbial ecosystems that we study. For this purpose, we use various specialist statistical methods and data visualization techniques. And as a sort of side project during my PhD, I developed a software toolkit for microbiome data science, aiming to make some of these specialist methods easier for other scientists to use. And this, I think, has been a rewarding and impactful side project, with thousands of scientists now using the MicroVis R package in hundreds of projects spanning many interesting domains of research. So, in conclusion, it's clear that dietary oligosaccharides are important for nurturing the developing infant gut microbiome. It's also clear that this developing microbiome is vulnerable to harm from antibiotic-induced collateral damage. But both galacto-oligosaccharides and the human milk oligosaccharide, 2FL, showed some potential in our experimental models in making the microbiome more functionally resilient. I think it's safe to say that having some oligosaccharides is likely always better than having no oligosaccharides. However, there's still much more we have to learn before we fully understand the extent to which the many different human milk oligosaccharides could have different effects on microbiota development or activities. But our research studies, along with others, have identified the real relevance of the infant gut microbiome in several aspects of child health, including in gastrointestinal well-being, but also in asthma, which is discussed more in the thesis chapter 6. And given this relevance to child health, it is important that we do build further on this kind of work so we can understand more precisely how we can reliably promote and protect healthy gut microbiome development in all infants. And in our future efforts to achieve this, it will be helpful to both make use of and to contribute to the latest developments in laboratory and computational methods. And with that, I thank you all for your attention, and I give the word back to the Pro-Rector.
Thank you for that beautifully clear presentation and also the rather charming PowerPoint slides, I must say. The opposition will now be opened by Professor Daisy Jonkers, who is Professor of Intestinal Health at Maastricht University and who served as chair of the assessment committee.
Thank you very much. Dear candidate, I would like to congratulate you and your supervisory team with really a beautiful and very interesting thesis. Very well written, but of course you're a native English speaker, so that helps. So I also learned a lot of your nice language there. And I also have a lot of appreciation for the fact that you did develop this visualization tool and statistical tool, which I think is really helpful in this era of growing microbiome studies. So really very nice, and also all the publications you had. So I was triggered by a lot of findings, and I think it's very relevant to understand which factors affect the microbiome development in early life, as you also nicely illustrated. And in chapter 3, you used the koala birth cohort, and you actually confirmed nicely the impact of breastfeeding, antibiotic exposure, and also a lot of other factors there. But you also concluded, as you also mentioned in your presentation, that we do not know everything yet. In a further study, you showed impact of the HMOs, but that was also only modest. So my key question to understand the microbiome development is, what else? What could be other factors we should consider in future research, maybe to better understand the development of the microbiome? What are your thoughts about that?
So, dear highly esteemed opponent, thank you very much for your kind words and your interesting question to start us off. So, indeed, at this early time point in infant development, there is quite a lot also, I think it's important to note, of stochastic variation in between individuals. So I think we'll never get to the point where we can fully explain all aspects of this process, all aspects of differences between infants, particularly at this early time point. But I think that we can definitely get further than we are now. I think that in terms of other factors that are relevant for microbiome composition at this early age, besides the feeding mode, the potential antibiotics exposure, I think that later on in infancy, we're going to be seeing the influence of varied complementary feeding aspects. I think that will be particularly important later on in infancy. I think that the early environment, so thinking still about this very early age, I think the environmental microbes that individuals come into contact with, which are largely human microbes spread into the environment that we pick back up again, whether that be from the hospital or home environment. We did, for example, also find an impact of vaginal delivery at home versus at hospital was slightly influencing the microbiome at this time. So I think that that aspect of where we're obtaining these first microbes from is probably going to be helpful to piece.
Yeah, so you think I maybe further go into the exposome, the environmental factors. What about host genetics? I always wonder, because also in the adult you see like about, depends on the publication, 15% is explained by all the factors we know. So I think, do we maybe underscore host genetics here? What are your thoughts?
Yeah, so there have been some substantial studies in adults primarily on the influence of host genetics on microbiome variation. And whilst they do find some associations with various loci, they are largely pretty small effects there. But one thing I think is potentially also interesting to note here, because one mechanism of genetic influence could be through immune system variation. But another aspect that seems relevant to bring up at this point is also the exposure history to various microbes of the mother and the way this could be passed down to the infant through immunoglobulin A and that sort of thing that comes in through the breast milk. And this may have some quite important effects, I think, I would suspect, in the first microbes that actually start engrafting. And
No, I think very, very interesting for the future. Luckily, there's still a lot to do. I'm also very intrigued by chapter 5, where you really dive into the infant health and have this very interesting association with the distress of the mother and then the symptoms of the child, for example, the colic. So I'm just wondering, because it was quite a clear association you found, so I was really wondering how to understand the maternal distress and how that could impact the microbiome of the child. So what are your thoughts about that? How could that work? Or is it just a coincidental finding?
So the primary thing we identified there in chapter 5 with regards to the maternal distress score at 2 weeks was the strong positive associations, or consistent at least positive associations, with the symptoms in the child. Whether or not this is directly affecting the microbiome of the child was not an aspect we directly considered there, but it is kind of implicit in our use of this as a covariate in our adjusted statistical models. And I think that it is, so there's two parts there, I guess, of that question. One, what could be the mechanism of the mother to child kind of stress to child symptoms, and I think that's multifold. One, it could be a reporting bias in the sense that mothers who are experiencing already an aspect of higher stress may be more likely to experience their child's symptoms as being worse, because it is a self-report thing. But it could also reflect, perhaps, I think, a shared propensity for a kind of gut-brain axis dysfunction, perhaps, where you may have a higher propensity to anxiety, and the infant may have essentially be expressing a anxiety or unrest-like picture already early in life. Many mechanisms.
Now you really come to my field of interest, but I'm looking to the prorector whether there's tiny bit of time left, or I really have to choose the question I'll ask. I would like to go to proposition 5. Because of time, I will not read it out loud, but you say that statistics are needed to design a study. Now, you use the koala bird cohort. If you could, as a statistician, could redesign the bird cohort, what would you do to answer your questions, for example, which you tried to answer in chapter 5? Can you briefly elaborate on that?
Most helpful thing would have been more time points of microbiome composition, I think. At this scale, with more time points, that would have been, I think, brilliant to really unpick the temporal nature of the associations here. I would also like, perhaps, a different way of assessing symptoms, perhaps some kind of experience sampling-based method for the mother reporting, perhaps slightly more objectively, symptoms, frequency and severity in the child. I think those would be aspects I would really like to see.
I think given the time, we will discuss it further during the reception. Thank you very much for your answers. I'm very happy with them.
Thank you.
Thank you, Professor Jonkers. The opposition will now be continued by Dr. Henrik Munch-Roge, Associate Professor of the Microbiome and Metabolomics at the University of Copenhagen, and we're very grateful that you have come all the way here.
Thank you very much. Dear candidate, I really enjoyed reading your thesis, and I have a few questions I would like to ask. So, starting with the HMOs, you measured in breast milk. Could you have done it in feces, and would that have added any value to your story?
Dear esteemed opponent, thank you for your kind words and your question. So, measurement of HMOs in feces had actually been done in a smaller subset that was published previously of some of these same infants. And here you could assess the capacity of the gut microbes in the infant to degrade each of the different types of HMOs here. I think that would be the primary aim or interest of doing this. But in this previous work, it was interesting. You could associate the different degradation patterns with, to some extent, the different microbial communities in the different infants. But there were challenges there in the quantification in breast milk relative to in the feces that kind of prevented a kind of really quantitative assessment of how much degradation could have happened. So I think if I'm recalling correctly from that study, it was split into like high, medium, and low kind of levels of degradation.
So talking about quantification, you also ended up looking at in terms of relative abundances of the different HMOs. So you had 18 different HMOs, and then you did some relative proportions. And then I noticed that in one of your studies you had free FL, and another study you didn't have free FL. So what would that mean for the relative proportions of HMOs or for the data analysis and interpretations?
Indeed. So in the koala study, we had some issues with the measurement of 3FL and the reliability of those measurements, as it was done with a different technique. And so that couldn't, unfortunately, be included in those analyses. I do think that it would be, to some extent, impactful here, because in the non-secretors, you would expect a substantial proportion of 3FL essentially in place of the 2FL that they would lack. And so I think this could have some influence here on the results that we obtain there. But I did think a bit about this and was doing a bit of a sensitivity analysis with the more quantitative or absolute quantitative, as far as we could get that information, HMO abundances, and saw similar results. So I don't think it had a huge impact there. But
Okay. Then moving to the colic study, you find associations to Staphylococcus with persistent constipation. So Staphylococcus, where would you naturally find such, where does this bacteria come from?
I suspect one source of the Staphylococcus could be skin, potentially from around the mother's breast. Has also been identified as being part of the breast milk microbiome in theory, but it's hard to distinguish that from the skin microbiome. But yeah, so I suspect that this could be a skin microbe.
So how would you interpret this correlation you see between this bacteria and then their constipation?
So one thing we considered here is whether this was just reflecting reflecting breastfeeding history, essentially. But we still identified this association within the exclusively breastfed infants. But one thing
could it be frequency of breastfeeding? So the number of times because breastfeeding is not breastfeeding. It could also be number of breastfeedings.
Yeah. And I was thinking as well about this, that it might relate to mode of breastfeeding, whether it was pumped or direct on the breast, as that's been shown also to have an influence on the breast milk microbiome, as it were, and also you could imagine easily that that would affect the microbes the infant was ingesting. So there could still be a link there with more details about how breastfeeding was done.
And how about this Ruminococcus scinereus and its link to colicky symptoms? So what is, if you look in early life, what else have this bacteria been linked to?
I had noticed in the child study, a large Canadian birth cohort, that earlier appearance of Ruminococcus narvis had been linked to later atopic disease. I think it was asthma in that case, but I cannot remember exactly.
How about feeding mode?
Yeah, so in that case, I believe the infants who were more formula-fed or transitioned earlier to formula feeding where Ruminococcus narvis was appearing earlier in those infants.
Yeah, so it's very often seen higher in those that are formula-fed. Do you know why?
I might suspect that it's a competition issue with regards to the less likelihood of being outcompeted by bifidobacterium in this early stage, but perhaps there's some other...
It could also be the amount of amino acids and proteins that you find in their formula, which tends to be higher.
Okay.
Anyways, if you think about that, you have one marker that seems maybe to be linked to breastfeeding, and you have another marker which could be interpreted to be more linked to formula feeding. My question is just whether these symptoms are basically linked to feeding, and whether you check for this in your analysis.
Indeed. I think, so firstly, we did both do our analyses adjusted for the breastfeeding history of the child, and also analyses in the subgroup of breastfed infants to kind of assess this at least on the level of whether or not or how much the infant was breastfed. But I do think that you're getting to something I would agree with in terms of the exact details of how the feeding is occurring is still not, it still could essentially be residual confounding here of these associations. So it may still be a marker of feeding, potentially confounding this influence.
Okay. So I think I have time for one last question. Okay. So moving to your last chapter 7 and the in vitro models, you end up pooling donor material into your system, and you want to study ecology and cross-feeding, and you see some interesting things, but what are sort of the limitations of pooling the donors and, yeah?
Yes, so here we tried at least to mitigate the effects of pooling multiple infant species together, which we did in order to have sufficient material to use in the system. We attempted to mitigate kind of getting some of the problems with this by assessing the individuals' microbiome compositions and pooling together at least their compositions as far as we can assess with 16S sequencing, and pooling together ones that are most similar. But I think that one of the main limitations we get here is that I think that inter-individual differences in response will be hugely interesting to study here. It's just with this system we were unable to look at that. I think that artificially having more strains of the same species than you might normally have might be doing unusual things in this system, but that's not something we could assess.
Okay, thank you very much.
Thank you.
Thank you. The opposition will be continued by Professor Carolina de Weert, Professor of Developmental Psychobiology from the Radboud University Medical Center. And we also appreciate that you've made it all this way.
Thank you. So first, congratulations with your thesis. I want to extend my congratulations also to the promoters and to family and friends. They're also important in writing a thesis. And I really enjoyed it. I thought you had a very solid introduction, very pleasant to read, and strong statistics. That was appreciated. And your discussion I found very inspiring as well. So congratulations. But of course, I'm going to be very critical now. I have a chance to do it. So I wanted to ask you about chapter 5 and about the methodology used for colic. So I have some doubts about the way you asked mothers with questionnaires that covered a period of 3 months to report about how much their babies had cried, how often. Actually, you asked them about frequency, duration, how many hours. And we know that colic originates a couple of weeks after birth. It peaks at around 6 weeks, 7 weeks. And by 3 months, it's usually resolved. So you ask them when it was already resolved. And then you ask them again at 7 months and at 12 months, when I can imagine that there's much less crying going on in the infants during those periods. So how do you know their reports were reliable?
Dear highly esteemed opponent, thank you very much for your kind words and for your interesting question. So I completely agree that this is one issue that's very important to consider in this kind of research, where we have these self-report questions, and they're asking the parent to reflect over quite some time period retrospectively. I think that the major effect that this could have on our analyses would be biasing our statistics towards null, towards not finding associations here. So I think that is a concern, but one that I think would not be concerning in the sense that the associations we do find would be problematic. I wouldn't see that we would bias towards finding associations here necessarily that we wouldn't otherwise find.
How were parents told to diagnose cramps and constipation?
So these were very simple questions asking initially if their baby was suffering substantially from either constipation, obstipation, darmverstopping, and excuse my terrible Dutch, and if yes, for what length of time in weeks, and then for how frequently within that time period. And then the same for cramps. It was just asking if they were suffering from
Crampius.
From cramps, yeah, Darmcrampius, yeah.
Yeah.
So indeed, very simple kind of symptom reports there. So it's not really diagnostic per se, but it's still, I think, something we can use as...
But given that they had to remember a whole 3 months period, and you were asking them pretty detailed, I have some doubts about how specific and how, what people mean by crampius is very different, and constipation can also be very much related to diet, as was just asked before. Mm-hmm. So, and then I come to the part where you talk about the Rhaminococcus gnathus, what we were just discussing. And you said that it hadn't been noted in prior studies of infant colic. And that's actually not the case, because in our 2013 study, we found lower relative abundances of this bacterium at 2 weeks of age in infants with colic. So where you say that it seems to be a pretty coherent story in which Ruminococcus gnathus disrupts intestinal physiology, I would say that it doesn't seem the case. At least in our own study, we found that it was lower. So the opposite. How would you explain this?
So firstly, yes, I'm familiar with the study, but must have missed that particular detail indeed. So I think that one thing about Ruminococcus gnathus is that the strains of Ruminococcus gnathus seem to differ considerably in their immunogenicity, depending on whether or not they put a polysaccharide capsule around the pro-inflammatory carbohydrate. And so I start to wonder if associations that would be identified there would be dependent on all sorts of nuances with regards to the prevalence of certain strains within certain aspects of the population. So you could really get quite different associations depending on...
Not to go into so much detail on the strains, isn't there a more obvious difference in the way you asked about colic? You found the strongest results with the mother's reports at 7 months on their crying, when it's a period where they shouldn't be crying a lot. Colic is over. No colic at between 3 or 4 and 7 months. Usually you don't have colic, or it's not called colic. And we found our results at 2 weeks, so very early. Couldn't it have to do with this period? Could it be that you are actually not measuring colic, but that you're measuring persistent regulatory problems in these children? Because you're asking them at 7 months, did your baby cry a lot? And the babies that are crying a lot at 7 months, they may be that very small group of the population that does continue to have feeding problems, sleeping problems, and crying problems, regulatory problems. So maybe you're not talking about colic in this study, I wonder. Because of the way you asked the questions, and because of the results that you found, which are different also from...
I would very much agree with that also, actually. I think that I'm often trying to be quite careful in calling this colicky crying symptoms, in the sense that it seems quite difficult to believe that colic per se is all going to be one thing across all different infants. And indeed, this later time point where we found the most strong association between the 4 and 7 months window, it does seem very reasonable that this would reflect some subtype of pathophysiology that could be driving something different than normal early infancy colic here.
Do you have time for a short last question?
Yes, given all the hassle you had coming here.
So in your general discussion, it has nothing to do with this, but you say that there should be education on our software for analyzing data and for open science, and I couldn't agree more. I thought that was a great idea to educate students better. But then I was wondering what you think about pre-registration, which is another important feature of open science. And you mentioned avoiding p-hacking as a danger of these complex analyses, but you don't seem to have used it yourself, pre-registration as a way of avoiding p-hacking. And in general, I'm wondering, because the analyses are very complex for microbiota. If you pre-register them, don't you put yourself in a straitjacket? How do you think about these things?
Yes, I think that pre-registration, in my mind, is particularly important if you want to do more of a confirmatory sort of analysis, where you really have quite specific hypotheses that you really want to pin down, and if, where possible there, you would be able to decide up front on a particular analysis plan that would achieve that aim. But I think that much of the work that we do in microbiome analysis falls more on the exploratory side of things, where we're really, at least initially, likely in the future, this can and should, I think, change as we start to get more of a handle on what we're expecting to find or what we're really hypothesizing to find. So I think, pre-registration, interesting, not perhaps you can pre-register part of the analysis, specify up to which point you're sticking within a certain framework, and then exploratory thereafter, but clearly designated.
So transparency.
Yeah, I think that makes sense.
Thank you very much.
Thank you very much. The opposition will now be continued by Professor Edward Dompling, Professor of Early Diagnosis and Pediatric Pulmonology at the Maastricht University Medical Center.
Thank you, Mr. Prorector. Mr. Candidate, so congratulations. It was really a very nice thesis. It was lovely to read it, and I love your analysis, and I'm especially fond of your very nice figures. I was wondering whether, I suppose that these kind of figures are probably at your wallpaper at home. Maybe that's a good suggestion to start a commercial company in this. Maybe it's good in the new market. So I was really very enthusiastic about your thesis. And congratulations also to your team of supervisors. Also did a big job, I think, a great job. So but saying that, I have, of course, some questions. I'm a little bit concerned, actually, about the use of data of the koala study. Because the study is an excellent study, but it's already performed many years ago. Was it 5, 10, 15 years ago? So biosamples of stools were gathered for quite a long time in the freezer. What can you tell us about integrity of the So first, maybe how much time was there for the storage? Is indeed on average about 10 years. And what does that mean for the quality of the DNA in your samples? And how did you control for that?
So, dear highly esteemed opponent, thank you very much for your kind words and the suggestion about the wallpaper, and and for your question. As a further note on the wallpaper thing, whilst I don't have it as wallpaper, one figure that was particularly liked was submitted to a scientific illustration thing and was shortlisted, and so we do have a poster of that in the department.
I will come and look. Have a look. Yeah, thank you.
With regards to the DNA integrity over time, so the storage time was indeed almost closer to 20 years. But it was stored as isolated DNA rather than as the fecal samples themselves. And so with that, in minus 80 freezers. And so as far as I understand, the evidence should be that this is a pretty good long-term stable situation.
Do you have a kind of a comparison, that's data of stools who were immediately analyzed compared to the analysis nowadays? Of course, it's difficult because methods change in the course of time, but...
So we don't have a direct comparison there because where we would have comparisons, for example, with the baby carb study, the analyses were performed, or the laboratory analyses were performed at different laboratories, and that is likely to have also a very large technical effect on the microbiota that you would profile. So I think we didn't have an opportunity there to make that comparison directly here. But I believe that prior studies have done this and found good long-term stability.
Okay, so that's still something to be, to investigate, I think. Yeah. So can you read Proposition 1 for me, please?
I think that's paragraph duty.
Okay, doesn't matter. You talked already so long. So.
Typical oral antibiotic prescriptions during pregnancy do not significantly disturb neonatal gut microbiota assembly. This thesis.
Yeah, thank you. So, this were human data, and your discussion, you already said, it's a little bit in contradiction to the animal studies. So, can you explain briefly how can we explain that, the difference between these animal studies, where we found this kind of relationship, and the human study in children?
Yes, indeed. So there had been quite some concern and discussion in the literature and amongst medical professionals about whether the results that were obtained in mouse studies, where they fed quite high doses of antibiotics over extended periods of time to pregnant dams, this seemed to have a substantial influence both on the dam's microbiota, but also on the pup's microbiota. But I think the distinction and the concern was that this result could be translated to humans and might explain some of the associations that have been seen between pregnancy antibiotics in large cohorts and registry studies and higher rates of asthma in children born to those mothers. But I think the main difference here is likely in dose and timing and class of antibiotic that's being used.
Yeah. So has all this detail be registered by the koala study? So I could imagine that the length of or duration of antibiotic use of the mothers, or the type of antibiotic, the macrolides or the penicillium-derived substances, could really have an influence. Did you have that information?
Unfortunately not. So we only knew self-report whether the mother had oral antibiotics during pregnancy and at what month of pregnancy that was, but we didn't know what or for how long.
On the other hand, in fact, as a clinician, I'm not surprised at all, actually. So I want to reason a little bit with you about just the absence of a relationship. Is it so abnormal? Because during pregnancy, mothers can use antibiotics, but the intestines of the child are more or less sterile. After birth, they are colonized with all kinds of bacteria. For instance, mostly from the mother. Also dependent of the way of the delivery, cesarean section or normal birth. So actually, I'm not that much surprised about, didn't find an association between the two. So what's your opinion about that? Can you follow me there and that? And
yes, I think I would largely agree with you here that I also was not particularly surprised to not see an effect here. I think that if you were to see an effect on the 1 month microbiota, gut microbiota of these infants, the implication would have been that you'd have had enough of a substantial impact on the mother's gut microbiota that it was changed enough such that when microbes were transferring from the mother to the infant, either around birth or subsequently, that this would change the inoculum of the developing infant enough to change its microbiome. But so it doesn't surprise me particularly that that mechanism doesn't seem to be present in normal clinical practice.
Thank you. I think time is a little bit off. Still a short question. Let's speculate a little bit about Proposition 8, the potential of carbohydrate to promote child health by modulation. Can you speculate on that? What kind of substances or molecules are interesting? Or isn't it better to go to a short-chain fatty acid? Because that's more a molecule more in connection and more distal in the whole process of influencing the microbiome?
Yeah, it's an interesting question. So the question really relates, I guess, to whether you give the microbes what they...
Please, a brief answer.
Yes, what they need to feed to produce things like short-chain fatty acids, or whether you would directly give the short-chain fatty acids. And I think if you had a good mechanism to consistently give short-chain fatty acids in the distal colon in a non-invasive way, that would be a good option. But I think that lacking that, I think feeding the microbes is a more tractable strategy.
Well, maybe we can speculate about that in the future. Yeah. So I give the word back to the prorector. Thank you.
Thank you, Professor Doppeling. The opposition will now be continued by Dr. Susan Steinbusch-Koert, Associate Professor of Bioinformatics at Maastricht University.
Thank you. Yeah, dear candidate, also I want to congratulate you with a wonderful piece of work. I think you did a marvelous job in connecting different cohort studies, also combining model systems, and then with a big umbrella of computational advanced methods. So well done. And I also want to extend my congratulations to your supervisory team. I would also like to start with one of your propositions. I think were all very challenging. I think we all agreed upon that. So I would like to ask one of your paronyms to read proposition number 6.
Multi-omic analysis of complex host-associated microbiomes will present some of the greatest challenges yet seen in bioinformatics, biostatistics, and systems biology.
Yeah, that's a whole mouthful, I think, a lot of terms there, but I couldn't agree more. I was wondering, what type of omics data are you referring to, and what do you think are the most critical bottlenecks then in performing this integrative analysis?
Dear esteemed opponent, thank you very much for your kind words and for your interesting question. So, yeah, I think that in terms of which omics analyses I'm talking about, I think that the challenges associated with microbiome data here can extend across pretty much all of them, but I think the easiest one will, of course, be genomics, because then it's much more of a case of presence, absence, and abundance. But with further omics, you have issues of both the fact that microbes can be present and absent, dynamically changing over time, but then also, if you were to look at transcriptomics, metabolomics, proteomics, you have to somehow factor in the changes within the microbes that are either there or not there, or abundant or not abundant at any given time point, and what they are doing, their metabolic activity, their transcriptional regulation, all that sort of thing, all on these different timescales and sort of different levels in which these things can operate.
So I hear also that you need this temporal effect then to say something reasonable about that host microbiome interaction. But you would combine everything. So metatranscriptomics, metabolomics, proteomics, is not one you favor most, maybe, of combining with your metagenomics data.
I think this would hugely depend on the question you had and the type of system you were studying. I think that transcriptomics or metatranscriptomics is something where I think in experimental models, combining that with your genomics and maybe also metabolomics, you can make the link there between who exactly is making, so you have who's present, what they're making an aggregate, and you can start to get some idea with metatranscriptomics in a model system, the who is responsible, which microbes are responsible for which products in a more direct sense there. But yeah, and then in a human system, I think temporal regulation and in terms of having early enough access to RNA to the sample in order to get reliable, unbiased measurements of the RNA could be more challenging.
Yeah, that's also getting in the right sample. I agree. That's important as well. And then I was reading your discussion, and then you were also talking about some puzzle pieces that are still missing, I think, in the bioinformatics analysis, and you touched upon epigenetic regulation, which I haven't heard you talk about yet. And also that microbial activity could be influenced by epigenetic mechanisms. And now I wondered, because in the human breast milk, also the microbes that are in there, they also produce factors that could influence epigenetic. Is that something that you would be interested in? For example, looking at, we know, for example, that exomes that originate from these microbes can have microRNAs or enzymes that can influence methylation. Is that something that would also explain some of the effects that you see?
Yeah, that's a really interesting suggestion to think about. In the discussion, I was thinking, I believe, more on the lines of microbial epigenetics, so the epigenetics of the microbes themselves. But indeed, in terms of host epigenetic regulation by factors in breast milk, that would also be an interesting way of getting towards kind of transcriptional regulation without actually needing to measure the transcriptome itself. So that could be curious. But I think, yes, did I answer the question there?
Yeah, maybe it was more suggestion then. But have you ever then considered the exomes or
no, that's not something we considered up until this point.
Then I have a question related to chapter 3, where you looked at the genus taxa level, and you had a classification of these taxas across the different infants, and you also specifically looked at the breastfed infants. And there you do see some dominant taxa occurring. And we know from in adult situations that also if there's a dominant one, that they often classify that in enterotypes. And I know that's debatable, and also these enterotypes are then linked also to dietary intake. But I was wondering, could you already somehow classify your infants and maybe based on a classification get an insight into a specific health outcome later on?
Yeah, it's an interesting question, and I think that it would depend on which level of measurement you were using as to how useful I think these classifications or subgrouping or enterotyping, whatever you'd want to call it, could be. I think that at genus level, you might run the risk of these not being super reliable classifications, but I think that I personally feel that the concern with coming up with these kind of simplified summaries of an ecosystem is that you miss the point sometimes in terms of at least explanatory research. In some cases, if you had the right level of measurement and the groups represented something really biologically meaningful or reliably predictive.
So it also depends on the number that you can study. Is that also important then? Or?
Yeah, and I think also the populations you study, the generalizability of this kind of grouping. I think gut microbiome composition really varies geographically, for one thing. And so I think that if you were to build a model that would predict from simple enterotyping, certain disease risk in one population, the Dutch population, you might struggle to apply this to other populations, for example. But I think if you were to get closer to something more mechanistically interpretable, rather than something like enterotyping, you might get something more reliably generalizable, perhaps.
And what are you thinking of then, if you say more mechanistically?
So this could be things like capacity to produce certain short-chain fatty acids or indole lactate, or in some cases there are substances that you would not want the microbiome to be able to produce. So either you measure them directly, like there's a certain fatty acid, if I recall something, 1,2-dihome has been associated with a higher risk of asthma. If you started pinpointing these kind of things, then I think you've got something more predictive. Yeah. And more generalizable. Okay.
I think I agree with you there. I think that having just a snapshot is not sufficient. So that should, yes. Yeah, you already touched upon that you designed this microvis R package, and I just want to mention it's a really short chapter in your thesis, but I think it's a lot of work and it shouldn't be overlooked, and it was already addressed. It's something that has a great impact. I wanted to do something.
Thank you.
David Barnett, the time appointed for defending your thesis has now passed. The degree committee will now withdraw to discuss the quality of your thesis and of your defense. And I therefore request that you and your company await the results of our deliberation and our return in this room.
All right, please.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. The line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. road to the south side, 7 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side, no place like home. If there's questions that you've got, go the extra mile and die.
On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off.
Take the mileage, traveling so far from home, gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you won the battle once you've grown. South side, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side, no place like home. If it's questions that you've got, go the extra mile and die. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side, no place like home. If it's questions that you've got, go the extra mile and die.
Years can't fly by now, don't waste all your time. Cause I'll go the extra mile.
David Barnett, the degree committee here present today has assessed the quality of your thesis and of your defense. In view of its positive verdict and taking into account your previous qualification, the degree committee has decided to grant you the degree of doctor. Professor Aerts is authorized to confer upon you this academic distinction in accordance with Dutch university custom. And I now invite your supervisor to take the floor.
All right, please.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, David James Mark Barnett, the degree of doctor and grant you all the rights attached by custom and law. As evidence of this, I now present you with the degree certificate, signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university. Let me see if I can find this. Here it is.
Okay, now it's my turn. Dear David, dear Dr. Barnett, congratulations. You did a very well job, and that deserves a big congratulation. You really showed today how knowledgeable you are when it comes down to microbiome data analysis and the microbiome in infants. And of course, I would also like to extend my congratulations to Rebecca, also with the new wallpaper you soon get, and to the rest of your family with this major achievement. And having completed your PhD must feel great, especially since the final part of this journey had to be done in between all of the new responsibilities and projects as a bioinformatician in our department. Now, your outstanding academic performance was already apparent during your studies, for which you received several distinctions. First, at the medical school at Brighton in Sussex, where you left after 2 years to continue in biomedical sciences, and also received a degree with honors. And thereafter, you moved to Maastricht to follow the Masters of Health Science Research with a specialization in epidemiology, which you completed cum laude. And it was during this master where your journey in the microbiome field started, during an internship under the supervision of Dr. Karel Thijs. You explored the association between metagenic archaea in the gut microbiota of school-aged children and the risk of asthma. And from the very first start, you demonstrated how well organized you are as a data scientist. This is a great asset. And the paper was well received by the Journal of Allergy and Clinical Immunology, the #1 journal in the field, although you were actually a little bit upset because it was not accepted as a full paper, but as a letter. And at first you had some doubts about it because you had to abandon a lot of the data analysis you had to do. But being convinced that everything could be published as an online repository, I think that was really convincing to you. And I think that really shows also your scientific integrity. When the grant proposal led by Professor Ilya Aerts and in collaboration with the Wageningen University was funded by the Carbohydrate Competence Center partnership, we knew you would be an excellent candidate for the dry lab part of the project. You were appointed at the Maastricht Center for Systems Biology, and with Ilya as first promoter, but you were also very visible in our department. And I think this really shows a great example of inter-faculty and inter-institute collaborations. Moreover, within this project, you closely collaborated with Marta Endika, who is also present here today, and who was appointed as a PhD candidate in Wageningen under the supervision of Hauke Smit, and performed the in vitro studies using the TEM2 system in Koen Venema's lab. Although mostly remotely, the collaboration between the two of you was really smooth and a great added value for the project. You assisted in the data analysis of Marta's in vitro studies, and Marta jumped in to speed up the HMO profiling in the koala milk samples, and in this way both of you managed to minimize the delay as a result of the COVID pandemic. Within this NWO-CCC Carbohydrates Consortium, which is a public-private partnership, I would also like to thank all the partners for the nice and constructive collaborations. And you are actually the second PhD from this consortium that graduated this week. I would also like to thank all the members of the Defense Committee, and in particular Henrik Roger and Carolina de Weert for coming from Nijmegen and Copenhagen to be here on site with us today. But let's get back to you now. The fact that you were born to analyze the data and definitely did not belong in the lab became very soon clear to me. Within the first weeks of your PhD, we needed to select some samples to be shipped to Wageningen. And I remember well that Kessel, one of our research technicians, together with me, guided you through our labs to show where the samples were and how they should be allocated and shipped to Wageningen. It was utter fear that I saw in your eyes when you realized you needed to do some lab work, if only it was picking the samples. And I think you have never set any step in the lab ever since. But behind your computer you excelled. This was amongst others proven by the side projects you started during the pandemic, when the release of the actual data of your project was significantly delayed due to lab closures, and you suggested to build an R package for microbiome data and visualization, and it was also already mentioned before today. This was really an asset of your PhD. It is well cited and it's used by many people across the world. When asking about your skills and your personality, the responses were extremely consistent. You were very open to helping others in their data analysis and gifted in explaining complicated scripts and methods in an easy and understandable way. Some of the quotes were, "His coding skills are unmatched. His commitment to support research is unwavering no matter the time. His willingness to help even when the rest of the world seems to be asleep." And the latter relates to the fast response you give to people living in completely different time zones, even after office hours. Besides your support and good teaching qualities, there is one word that describes you, in my opinion, best: meticulous. I even think that in the dictionary, the explanation of this word should be David, as I've never seen anyone who has such great attention to detail, accuracy, and precision. And the progress meetings with Ilya and me were always well prepared with presentations, HTML data analysis reports, actions, and to-do lists. We hardly ever had to ask you for additional information. On the contrary, most of the time we needed to push you to leave some of the details out to make sure we finished in time. It is such a great pleasure to be able to fully rely on the accuracy of the data, although even you can make mistakes, and we found some in one of the papers that was published. And although minor and not affecting the conclusions, we submitted an erratum with a corrected analysis to the journal. And you were quite frustrated. The slow and laconic attitude of the journal to pick this up and to correct this. And again, I think that shows your scientific integrity. But your biggest strength can also be a weakness, as you are meticulous in everything, no matter the importance. When we went over to your slides earlier this month, I jokingly said to you, it looks like you included all the 1,000 participants illustrated as dots in the slide showing the study design. That's serious, you said. Yes, these are actually 1,000 dots on the slide. And also during our recent conference trip to Rome, where we shared an apartment with our group, it became clear that you are the epitome of organization. So meticulous that even Marie Kondo would throw the towel in. The downside of that was that we all had to wait at least half an hour before you were finally ready to get out. But this was well compensated by your presence, because you're not only highly appreciated for your skills, but also for your pleasant company during social activities. I'm therefore also very pleased that you will continue your career as part of our team to further extend the microbiome research, assist in all the projects, as well as in teaching, and I really look forward to this new phase in your career. Once again, congratulations, and now let's celebrate.
Dear Dr. Barnett, Also on behalf of Maastricht University, I would like to congratulate you with the degree you have acquired. A doctorate is, as they say, forever, and I hope that you will live up to the values embodied in it for as long as you continue to contribute to science. What happens now is that we will take some pictures at the stairs. I believe that the after party is in the rafter, so I invite you all to, once we have left, to go over there, and then we will join you very shortly to well, give you the opportunity to congratulate the new doctor. Thank you very much.